The human body is composed of approximately 30–40 trillion cells, though the exact number varies with body size, age, sex, and methodology used for estimation. These cells originate from a single fertilized egg and diversify through regulated processes of division and differentiation. Human cells are not uniform. They differ widely in size, shape, structure, lifespan, and function. More than 200 distinct cell types have been identified, each specialized to perform specific biological roles essential for survival. Cells are the smallest units of life capable of carrying out metabolism, growth, responsiveness, and reproduction. Every human cell is enclosed by a plasma membrane composed of a phospholipid bilayer, embedded with proteins, cholesterol, and carbohydrates. This membrane regulates the movement of ions, nutrients, signaling molecules, and waste products, maintaining internal chemical stability distinct from the external environment. Embedded receptors allow cells to detect and respond to chemical and physical signals. Most human cells contain a nucleus that houses genetic material in the form of DNA organized into chromosomes. These are classified as eukaryotic cells. Certain cell types, such as mature red blood cells, lack a nucleus in most organelles, a specialization that maximizes their capacity to transport oxygen. Other cells, such as skeletal muscle fibers, contain multiple nuclei resulting from the fusion of precursor cells during development. Within the cytoplasm, specialized structures called organelles perform defined tasks. Mitochondria generate adenosine triphosphate through oxidative phosphorylation and are present in numbers proportional to a cell's energy demand. Neurons and muscle cells may contain thousands of mitochondria, whereas cells with lower metabolic requirements contain fewer. Ribosomes synthesize proteins, either freely in the cytosol or bound to the rough endoplasmic reticulum. The smooth endoplasmic reticulum participates in lipid synthesis, calcium storage, and detoxification. The Golgi apparatus modifies, sorts, and packages proteins and lipids for transport or secretion. Cell size varies dramatically. Human sperm cells are among the smallest, while motor neurons can extend over a meter in length. Red blood cells have a biconcave shape approximately 7–8 micrometers in diameter, optimized for gas exchange and flexibility within capillaries. Oocytes are the largest cells in the human body by volume, containing abundant cytoplasmic reserves to support early embryonic development. Cell lifespan ranges from hours to decades. Intestinal epithelial cells are replaced every 3–5 days, skin epidermal cells every few weeks, and neutrophils survive only hours to days. In contrast, many neurons formed during early development persist for an entire lifetime, though they remain metabolically active and structurally plastic. Programmed cell death, or apoptosis, eliminates damaged, infected, or unnecessary cells in a controlled manner, preventing inflammation and maintaining tissue integrity. Cells do not function in isolation. They are organized into tissues through physical connections and chemical communication. Junctional complexes such as tight junctions, adherens junctions, desmosomes, and gap junctions mechanically link cells and permit selective exchange of ions and small molecules. The extracellular matrix, composed of proteins like collagen and elastin and polysaccharides such as proteoglycans, provides structural support and biochemical cues that influence cell behavior. Human cellular composition also includes a large population of non-human cells. The body hosts trillions of microorganisms, bacteria, archaea, fungi, and viruses, primarily residing on the skin, in the oral cavity, and within the gastrointestinal tract. Although genetically distinct from human cells, these organisms contribute to digestion, vitamin synthesis, immune system development, and protection against pathogens. By cell count, microbial cells are roughly comparable in number to human cells, though they contribute far less to total body mass. Cellular function is governed by gene expression patterns rather than differences in DNA sequence. Nearly all nucleated cells contain the same genome, but selective activation and repression of genes determine cellular identity. This regulation is achieved through transcription factors, chromatin remodeling, RNA processing, and post-translational modification of proteins. Cellular specialization is stable but not always irreversible. Under certain conditions, cells can change identity or be reprogrammed. The integrity of cellular processes depends on precise regulation of ion concentrations, pH, temperature, and energy availability. Disruption at the cellular level, through genetic mutations, metabolic failure, or external insults, can propagate to tissues and organs, ultimately manifesting as disease. The collective behavior of trillions of cells, each operating under biochemical and physical constraints, gives rise to the structure, function, and adaptability of the human body. The human genome consists of approximately 3.2 billion base pairs of DNA, organized into 23 pairs of chromosomes within the nucleus of most cells. 22 pairs are autosomes, and 1 pair constitutes the sex chromosomes. The genome encodes the instructions required for cellular structure, function, development, and maintenance. Despite its size, only about 1–2% of the genome directly codes for proteins. The remaining majority includes regulatory sequences, introns, non-coding RNA genes, repetitive elements, and structural regions essential for chromosome integrity and regulation. DNA is composed of 4 nucleotide bases, adenine, thymine, cytosine, and guanine, arranged in a double helix. The specific sequence of these bases encodes genetic information. Genes are discrete units of DNA that include coding regions called exons and non-coding regions called introns. During gene expression, introns are removed from the precursor messenger RNA through splicing, and exons are joined to form a mature mRNA molecule that is translated into a protein. Alternative splicing allows a single gene to produce multiple protein variants, significantly expanding the functional output of the genome. Chromosomes are not uniform strands of DNA. They are highly organized structures. DNA is wrapped around histone proteins to form nucleosomes, which further fold into higher-order chromatin structures. Chromatin exists in 2 primary states: euchromatin, which is relatively open and transcriptionally active, and heterochromatin, which is densely packed and generally transcriptionally silent. This structural organization influences which genes are accessible for transcription at any given time. Gene regulation refers to the mechanisms that control when, where, and how strongly genes are expressed. Regulation occurs at multiple levels, including chromatin modification, transcription initiation, RNA processing, mRNA transport, translation, and protein degradation. At the transcriptional level, regulatory DNA sequences such as promoters, enhancers, silencers, and insulators interact with transcription factors and cofactors to modulate gene activity. Enhancers can influence gene expression from distances of thousands of base pairs by looping through 3-dimensional chromatin architecture. Transcription factors are proteins that bind specific DNA sequences and either activate or repress transcription. Their activity is influenced by cellular signals such as hormones, growth factors, nutrient availability, and stress. Many transcription factors operate in networks, integrating multiple inputs to produce context-dependent gene expression patterns. The same transcription factor can regulate different genes in different cell types, depending on chromatin state and the presence of other regulatory proteins. Epigenetic mechanisms play a central role in genome regulation without altering the underlying DNA sequence. These include DNA methylation, histone modification, and chromatin remodeling. DNA methylation typically occurs at cytosine bases in CpG dinucleotides and is associated with long-term gene repression. Histone modifications, such as acetylation and methylation, alter chromatin accessibility and recruit regulatory proteins. Epigenetic marks are heritable through cell division and contribute to stable cellular identity. Non-coding RNAs are integral components of gene regulation. MicroRNAs bind complementary sequences in messenger RNAs, leading to translational repression or degradation. Long non-coding RNAs participate in chromatin modification, transcriptional control, and nuclear organization. Ribosomal RNA and transfer RNA are essential for protein synthesis, while small nuclear RNAs are required for RNA splicing. The genome encodes thousands of non-coding RNA molecules with diverse regulatory functions. Gene expression is highly dynamic and responsive to internal and external conditions. Environmental factors such as temperature, oxygen availability, toxins, nutrition, and mechanical stress can alter gene expression patterns through signaling pathways that converge on the genome. Developmental stage also strongly influences gene activity, with tightly regulated temporal sequences of gene expression guiding cell differentiation and organ formation. The genome is not static over a lifetime. DNA damage occurs continuously due to replication errors, metabolic byproducts, and environmental exposures. Cells possess multiple DNA repair mechanisms to maintain genomic integrity, including base excision repair, nucleotide excision repair, mismatch repair, and double-strand break repair. When damage is irreparable, cells may undergo cell cycle arrest or programmed cell death to prevent propagation of mutations. Although nearly all nucleated cells contain the same genome, differential gene regulation results in the vast diversity of cell types and functions in the human body. This selective use of genetic information enables specialization while preserving a common genetic blueprint. Disruption of genome structure or gene regulation can impair cellular function and underlies many diseases, reflecting the central role of regulated gene expression in human biology. Epigenetics refers to heritable and reversible changes in gene activity that do not involve alterations in the DNA sequence itself. These changes regulate how genetic information is accessed and used by cells, allowing organisms with identical genomes to develop different cell types, functions, and phenotypes. Epigenetic regulation is essential for normal development, cellular differentiation, and the maintenance of tissue-specific gene expression throughout life. One of the primary epigenetic mechanisms is DNA methylation. This process involves the addition of a methyl group to the cytosine base, most commonly at cytosine guanine dinucleotides. High levels of DNA methylation in gene promoter regions are generally associated with transcriptional repression. DNA methylation patterns are established during early development and are maintained during cell division by specialized enzymes, ensuring stability of cell identity. However, these patterns can be modified in response to internal signals and environmental influences. Histone modifications constitute another major epigenetic mechanism. DNA is wrapped around histone proteins to form nucleosomes, and the chemical modification of histone tails influences chromatin structure and gene accessibility. Common histone modifications include acetylation, methylation, phosphorylation, and ubiquitination. Histone acetylation is typically associated with open chromatin and active transcription, while certain types of histone methylation can either activate or repress gene expression, depending on the specific amino acid residue modified. These modifications act as signals that recruit regulatory proteins to chromatin. Chromatin remodeling complexes use energy from ATP to reposition, eject, or restructure nucleosomes. This alters the physical accessibility of DNA to transcription machinery without changing nucleotide sequences. Chromatin remodeling is critical during processes such as DNA replication, repair, and transcriptional activation or repression. Together with DNA methylation and histone modification, chromatin remodeling defines the functional state of genomic regions. Environmental factors can induce epigenetic changes that influence gene expression. Nutrition affects the availability of methyl donors such as folate and methionine, which are required for DNA and histone methylation. Exposure to toxins, pollutants, heavy metals, and certain pharmaceuticals can alter epigenetic marks by interfering with regulatory enzymes. Physical factors such as temperature, hypoxia, and mechanical stress also influence epigenetic states through cellular signaling pathways. Psychological and physiological stress can modify epigenetic patterns, particularly in genes involved in hormonal regulation, immune function, and neural activity. Stress-related hormones activate intracellular signaling cascades that converge on chromatin-modifying enzymes, leading to long-lasting changes in gene expression. These changes can affect behavior, metabolism, and disease susceptibility without altering DNA sequences. Epigenetic regulation plays a crucial role during embryonic development. Early embryonic cells undergo extensive epigenetic reprogramming, erasing many existing marks and establishing new ones that guide differentiation into specific tissues. Errors in epigenetic programming during development can disrupt normal organ formation and result in congenital abnormalities. Genomic imprinting is a specialized epigenetic phenomenon in which certain genes are expressed from only one parental allele due to parent-specific methylation patterns. Some epigenetic modifications are stable over many cell divisions, while others are transient and reversible. This flexibility allows cells to respond rapidly to changing conditions while preserving long-term identity. In certain cases, epigenetic states can persist across generations through germ cells, allowing environmental influences experienced by parents to affect gene expression patterns in offspring. The extent and mechanisms of such transgenerational inheritance in humans remain an area of active research. Epigenetic dysregulation is associated with numerous diseases. Abnormal DNA methylation patterns and histone modifications are observed in many cancers, where tumor suppressor genes may be silenced and oncogenes activated. Epigenetic alterations also contribute to metabolic disorders, autoimmune diseases, neurodevelopmental conditions, and age-related functional decline. Because epigenetic changes are potentially reversible, they are targets for therapeutic intervention. Aging is accompanied by gradual changes in epigenetic patterns across tissues. These include global decreases in DNA methylation, localized hypermethylation of specific genes, and altered histone modification profiles. Such changes affect genome stability, gene regulation, and cellular function, contributing to increased disease risk with age. Epigenetic markers are also used to estimate biological age more accurately than chronological age. Epigenetics provides a molecular link between the genome and the environment. By regulating gene activity without altering genetic code, epigenetic mechanisms enable adaptability, specialization, and long-term regulation of cellular behavior. The cumulative epigenetic state of cells reflects both inherited programming and lifelong environmental exposure, shaping human physiology and health. Stem cells are undifferentiated or partially differentiated cells capable of both self-renewal and differentiation into specialized cell types. These two defining properties distinguish stem cells from other cells in the human body. Self-renewal allows stem cells to divide repeatedly while maintaining their undifferentiated state, and differentiation enables them to generate one or more specialized cell lineages under appropriate biological conditions. Stem cells are classified according to their developmental potential. Totipotent cells, present only at the earliest stages following fertilization, can give rise to all embryonic and extraembryonic tissues. Pluripotent stem cells can differentiate into cells derived from all three germ layers, ectoderm, mesoderm, and endoderm, but cannot form extraembryonic structures. Multipotent stem cells have a more limited range and typically generate cell types within a specific tissue or organ system. Unipotent progenitor cells produce only one cell type, but retain the ability to self-renew. Embryonic stem cells are derived from the inner cell mass of the blastocyst during early development. These cells are pluripotent and exhibit high proliferative capacity. Their gene expression profiles maintain an undifferentiated state through tightly regulated transcriptional networks. Embryonic stem cells can differentiate into virtually all cell types of the human body under controlled conditions, making them valuable for developmental biology research. Adult stem cells, also known as somatic stem cells, are found in developed tissues and serve as reservoirs for maintenance and repair. They are typically multipotent and restricted to generating cell types relevant to their tissue of origin. Examples include hematopoietic stem cells in bone marrow, which produce all blood cell lineages, and epithelial stem cells in the skin and intestinal lining, which replenish rapidly turning over tissues. Adult stem cells divide less frequently than embryonic stem cells and are regulated by local microenvironments called stem cell niches. The stem cell niche consists of surrounding cells, extracellular matrix components, signaling molecules, and physical factors that regulate stem cell behavior. Signals from the niche control whether stem cells remain quiescent, self-renew, or begin differentiation. Disruption of niche signaling can impair tissue maintenance or lead to abnormal cell proliferation. Mechanical forces, oxygen concentration, and nutrient availability within the niche also influence stem cell fate decisions. Cellular differentiation is the process by which stem or progenitor cells acquire specific structural and functional characteristics. Differentiation involves coordinated changes in gene expression, chromatin structure, metabolism, and cell morphology. These changes are driven by signaling pathways activated by growth factors, cell-cell interactions, and extracellular matrix cues. Once differentiation reaches a terminal stage, most cells lose the capacity to divide or revert to a stem-like state under normal physiological conditions. Differentiation follows hierarchical pathways. Stem cells give rise to intermediate progenitor cells that undergo limited rounds of division before differentiating further. This hierarchy allows efficient production of large numbers of specialized cells while preserving a smaller population of stem cells. In tissues with high turnover, such as blood and intestinal epithelium, this system ensures continuous renewal without exhausting the stem cell pool. Induced pluripotent stem cells are generated by reprogramming differentiated adult cells back into a pluripotent state through the introduction of specific transcription factors. These cells closely resemble embryonic stem cells in morphology, gene expression, and differentiation potential. Reprogramming involves extensive epigenetic remodeling, including changes in DNA methylation and histone modification. Although induced pluripotent stem cells do not occur naturally, they demonstrate that cellular identity is not permanently fixed. Stem cell function is tightly regulated to prevent errors. Uncontrolled self-renewal or failed differentiation can result in abnormal growth. Many cancers exhibit stem cell-like properties, including the ability to self-renew and generate heterogeneous cell populations. Cancer stem cells are thought to contribute to tumor initiation, progression, and resistance to therapy, highlighting the importance of regulatory control in normal stem cell biology. During development, stem cells drive the formation of all tissues and organs through precisely timed differentiation programs. In adulthood, stem cells maintain tissue homeostasis and enable repair following injury. The capacity for regeneration varies among tissues and is largely determined by the availability, potency, and regulation of resident stem cell populations. Stem cells decline in number and functional capacity with age. Accumulated DNA damage, epigenetic drift, changes in niche signaling, and metabolic alterations contribute to reduced self-renewal and differentiation efficiency. This decline affects tissue maintenance and increases susceptibility to degenerative diseases. Stem cell biology provides a framework for understanding how complex multicellular organisms develop, maintain structure, and respond to damage through regulated cellular differentiation. Human tissues are groups of cells with similar structure, origin, and function, organized to perform specific physiological roles. The human body contains 4 fundamental tissue types: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Each tissue type arises from one or more embryonic germ layers and exhibits characteristic cellular organization, extracellular components, and regenerative capacity. Epithelial tissue forms continuous sheets of closely packed cells that cover body surfaces, line cavities, and form glands. These cells exhibit polarity, with distinct apical and basal surfaces, and are anchored to an underlying basement membrane composed of collagen, laminin, and proteoglycans. Epithelial tissues lack blood vessels and rely on diffusion for nutrient and oxygen supply. They display high rates of cell turnover due to constant exposure to mechanical stress, pathogens, and chemical agents. Stem cells located in the basal layers continuously replace lost or damaged epithelial cells. Connective tissue provides structural support, protection, binding, and transport. It includes diverse subtypes such as bone, cartilage, adipose tissue, blood, and fibrous connective tissue. Connective tissues are characterized by the presence of an extracellular matrix composed of fibers and ground substance produced by resident cells. Collagen fibers provide tensile strength, elastin fibers allow stretch and recoil, and reticular fibers form supportive networks. The regenerative capacity of connective tissue varies widely depending on vascularization and cell turnover. Bone is a specialized connective tissue that provides mechanical support, protects vital organs, stores minerals, and houses hematopoietic stem cells. Bone tissue is dynamic and undergoes continuous remodeling through the coordinated activity of osteoblasts, osteoclasts, and osteocytes. This remodeling allows adaptation to mechanical stress and maintenance of mineral homeostasis. Bone regeneration following fracture is generally effective due to good blood supply and the presence of osteoprogenitor cells. Cartilage is a connective tissue with a firm but flexible matrix, composed primarily of collagen and proteoglycans produced by chondrocytes. It is avascular and receives nutrients by diffusion, which limits its regenerative capacity. Damage to cartilage, particularly articular cartilage in joints, heals slowly and often incompletely. Fibrocartilage and elastic cartilage also exhibit limited repair capabilities due to low cellularity and restricted nutrient access. Adipose tissue stores energy in the form of triglycerides, provides insulation, and functions as an endocrine organ by secreting hormones and signaling molecules. Adipocytes can increase or decrease in size in response to energy balance. Adipose tissue has moderate regenerative capacity through the differentiation of precursor cells, allowing expansion or reduction of fat mass over time. Blood is a connective tissue composed of cells suspended in plasma. Blood cells are continuously replenished from hematopoietic stem cells in the bone marrow. Red blood cells, white blood cells, and platelets have defined lifespans and are replaced at high rates. Blood exhibits one of the highest regenerative capacities in the human body due to ongoing stem cell activity. Muscle tissue is specialized for contraction and force generation. There are 3 types: skeletal, cardiac, and smooth muscle. Skeletal muscle fibers are long, multinucleated cells capable of voluntary contraction. These fibers have limited regenerative ability, relying on satellite cells for repair following injury. Cardiac muscle cells are highly specialized, electrically coupled, and responsible for rhythmic heart contractions. They have minimal regenerative capacity, and damage to cardiac tissue is typically replaced with fibrous connective tissue. Smooth muscle cells line hollow organs and blood vessels and retain greater capacity for proliferation and repair than skeletal and cardiac muscle. Nervous tissue is composed of neuron and supporting glial cells. Neurons transmit electrical and chemical signals, while glial cells provide structural support, insulation, metabolic assistance, and immune defense within the nervous system. Neurons in the central nervous system have very limited regenerative capacity after injury, largely due to inhibitory molecular environments and restricted cell division. In contrast, certain glial cells can proliferate, and limited neurogenesis persists in specific regions of the adult brain. Tissue regeneration depends on factors such as stem cell availability, vascular supply, extracellular matrix integrity, and inflammatory responses. Well-vascularized tissues with active stem cell populations regenerate more effectively than avascular or highly specialized tissues. Chronic inflammation, aging, and metabolic disorders impair regenerative processes by disrupting cellular signaling and tissue architecture. Scar formation is a common outcome when regeneration is incomplete. Fibrous connective tissue replaces damaged structures, restoring mechanical stability, but often reducing functional capacity. The balance between true regeneration and scarring varies by tissue type and injury severity. Human tissues exhibit a spectrum of regenerative abilities, from rapid renewal in epithelia and blood to limited repair in cartilage and nervous tissue. This variation reflects differences in cellular turnover, structural complexity, and evolutionary constraints, and it plays a central role in health, recovery from injury, and the progression of degenerative diseases. The human skeletal system consists of 206 bones in the adult body, though infants are born with approximately 270 bones that later fuse during growth. The skeleton provides structural support, protects vital organs, enables movement through leverage, stores minerals, and houses bone marrow for blood cell production. Bones vary widely in size, shape, and internal structure depending on their function and mechanical demands. Bones are classified by shape into long, short, flat, irregular, and sesamoid types. Long bones, such as the femur and humerus, function primarily as levers for movement. Short bones provide stability with limited motion, while flat bones protect organs and offer broad surfaces for muscle attachment. Irregular bones have complex shapes adapted to specialized roles, and sesamoid bones develop within tendons to reduce friction and modify force transmission. Bone tissue is a specialized form of connective tissue composed of cells embedded in a mineralized extracellular matrix. The matrix consists primarily of collagen fibers, which provide tensile strength, and hydroxyapatite crystals, which confer rigidity and resistance to compression. This composite structure allows bones to be strong yet relatively lightweight. There are two main types of bone tissue: compact bone and spongy bone. Compact bone forms the dense outer layer of bones and is organized into cylindrical units called osteons, which contain concentric layers of mineralized matrix surrounding central canals that house blood vessels and nerves. Spongy bone consists of a lattice-like network of trabeculae that reduces weight while maintaining strength and houses bone marrow. Bone cells include osteoblasts, osteocytes, and osteoclasts. Osteoblasts are responsible for synthesizing new bone matrix and initiating mineralization. Osteocytes are mature bone cells that reside in lacunae within the matrix and maintain bone tissue by sensing mechanical stress and regulating mineral exchange. Osteoclasts are large, multinucleated cells that break down bone tissue through enzymatic degradation and acidification, enabling remodeling and mineral release. Bone development occurs through two primary processes: intramembranous ossification and endochondral ossification. Intramembranous ossification forms flat bones directly from mesenchymal tissue. Endochondral ossification involves the replacement of a cartilage model with bone and is responsible for the formation of most long bones. These processes begin during embryonic development and continue into adolescence. Bone growth in length occurs at epiphyseal growth plates composed of cartilage located near the ends of long bones. Chondrocytes within these plates proliferate, mature, and are replaced by bone tissue, allowing bones to elongate. Growth plates close after puberty, when cartilage is fully replaced by bone, marking the end of longitudinal growth. Bones are dynamic tissues that continuously undergo remodeling throughout life. Remodeling involves the coordinated resorption of old bone by osteoclasts and formation of new bone by osteoblasts. This process repairs microdamage, adapts bone structure to mechanical loading, and maintains mineral homeostasis. Mechanical stress stimulates bone formation, while reduced loading leads to bone loss. Bones serve as major reservoirs for minerals, particularly calcium and phosphorus. Approximately 99% of the body's calcium is stored in bones and teeth. Calcium ions are released into the bloodstream when needed for physiological processes such as muscle contraction, nerve signaling, and blood clotting. Bone remodeling plays a critical role in regulating circulating mineral levels. Bone marrow exists in two forms: red marrow and yellow marrow. Red marrow is responsible for hematopoiesis, producing red blood cells, white blood cells, and platelets. It is abundant in infants and children and persists in specific adult bones such as the pelvis, ribs, sternum, and vertebrae. Yellow marrow primarily stores fat and can revert to red marrow under conditions of increased blood cell demand. The skeleton interacts closely with other body systems. Muscles attach to bones via tendons, enabling movement. Ligaments connect bones at joints, providing stability. Blood vessels and nerves traverse bone tissue to supply nutrients and sensory input. The skeletal system also influences endocrine function through the release of signaling molecules that affect metabolism and energy regulation. Bone strength and density are influenced by genetics, nutrition, hormonal balance, and mechanical loading. Adequate intake of calcium, phosphorus, and vitamin D supports bone mineralization. Hormones regulate bone turnover by modulating the activity of bone cells. Aging is associated with changes in bone remodeling balance, often resulting in reduced bone mass and increased fracture risk. The skeletal system is not a static framework but a living, adaptive structure. Its ability to grow, remodel, repair damage, and regulate mineral balance reflects the complex cellular and biochemical processes that sustain human movement, protection, and physiological stability. Bone remodeling is a continuous, lifelong process in which old or damaged bone tissue is removed and replaced with new bone. This process maintains skeletal strength, repairs microdamage, adapts bone structure to mechanical stress, and regulates mineral homeostasis. In healthy adults, approximately 5–10% of total bone mass is renewed each year, though rates vary by age, bone type, and physiological conditions. Remodeling occurs in discrete units, known as basic multicellular units. Each unit follows a coordinated sequence: activation, resorption, reversal, and formation. During activation, signaling molecules recruit osteoclast precursors to specific bone surfaces. In the resorption phase, osteoclasts attach to bone, create a sealed resorption zone, and dissolve mineralized matrix using acid and proteolytic enzymes. This phase typically lasts weeks. The reversal phase involves preparation of the bone surface for new deposition. During formation, osteoblasts synthesize new collagen matrix and promote mineralization, a process that may take several months. The balance between bone resorption and bone formation determines overall bone mass. When formation equals resorption, bone mass remains stable. When resorption exceeds formation, bone mass decreases. When formation exceeds resorption, bone mass increases. This balance is tightly regulated by mechanical forces, hormonal signals, nutritional status, and local cellular interactions. Mechanical loading is a primary determinant of bone remodeling. Bones subjected to regular stress become denser and stronger, while reduced loading leads to bone loss. This relationship is mediated by osteocytes, which function as mechanosensors. Osteocytes detect strain within the bone matrix and release signaling molecules that regulate osteoblast and osteoclast activity. Prolonged immobility or reduced gravitational load results in rapid bone loss due to decreased formation and increased resorption. Bone growth occurs through distinct mechanisms during development and adolescence. Longitudinal growth takes place at epiphyseal growth plates, where cartilage is progressively replaced by bone. Appositional growth increases bone diameter through the addition of new bone tissue on the outer surface, while resorption occurs on the inner surface to maintain appropriate thickness and shape. Peak bone mass is typically achieved in early adulthood, after which remodeling maintains but does not increase total bone mass. Bone mineral density reflects the amount of mineral per unit volume of bone and is a key determinant of bone strength. Density varies among skeletal sites and individuals and is influenced by genetics, physical activity, nutrition, and endocrine factors. Trabecular bone, which has a high surface area and rapid turnover, responds more quickly to metabolic and hormonal changes than compact bone. Mineral storage is a central function of bone. Calcium and phosphorus are deposited in the bone matrix as hydroxyapatite crystals, providing a readily accessible reserve. Blood calcium concentration is tightly regulated within a narrow range. When circulating calcium levels decrease, bone resorption increases to release calcium into the bloodstream. When calcium levels are sufficient or elevated, mineral deposition is favored. This exchange allows bones to serve as a dynamic mineral reservoir rather than a fixed storage site. Bone remodeling is closely integrated with systemic physiology. Hormonal signals influence the activity and lifespan of bone cells, thereby regulating turnover rates. Nutritional factors such as adequate protein, calcium, and vitamin D are required for effective bone formation and mineralization. Deficiencies impair osteoblast function and reduce bone strength. Aging alters bone remodeling dynamics. With advancing age, osteoblast activity and number decline, while osteoclast-mediated resorption often remains the same or increases. This shift leads to gradual loss of bone mass and deterioration of bone microarchitecture. Reduced osteocyte viability and altered signaling also impair the bone's ability to respond to mechanical loading. Microdamage accumulates in bone as a result of repeated stress. Remodeling targets these damaged areas, removing weakened tissue and replacing it with new bone. If remodeling is insufficient or imbalanced, microcracks can propagate, increasing fracture risk. Efficient remodeling is therefore essential for maintaining skeletal integrity under continuous mechanical demands. Bone growth, remodeling, and mineral storage are interdependent processes that allow the skeleton to function as a supportive, adaptive, and metabolically active organ system. Through coordinated cellular activity and precise regulation, bone maintains structural stability while simultaneously serving as a reservoir for essential minerals required by the rest of the body. Joints are anatomical structures where two or more bones meet, allowing movement and providing mechanical stability. The human body contains over 300 joints, varying in structure and function depending on location and mechanical demands. Joints are classified structurally as fibrous, cartilaginous, or synovial, and functionally by the degree of movement they permit. Fibrous joints are connected by dense connective tissue and allow little or no movement. Examples include sutures of the skull, which provide rigid protection for the brain, and syndesmoses, where bones are joined by ligaments or interosseous membranes. These joints are designed primarily for stability rather than mobility. Cartilaginous joints connect bones using cartilage and allow limited movement. In synchondroses, bones are joined by hyaline cartilage, such as the epiphyseal growth plates in developing bones. Symphyses involve fibrocartilage, as seen in the intervertebral discs and pubic symphysis. Cartilaginous joints provide strength and flexibility while absorbing compressive forces. Synovial joints are the most common and mobile joint type in the human body. They are characterized by a joint cavity filled with synovial fluid, articular cartilage covering bone ends, and a surrounding joint capsule. The capsule consists of an outer fibrous layer for stability and an inner synovial membrane that produces synovial fluid. Synovial fluid reduces friction, supplies nutrients to cartilage, and facilitates smooth movement. Articular cartilage is a specialized form of hyaline cartilage that covers the surfaces of bones in synovial joints. It is composed primarily of water, collagen fibers, and proteoglycans produced by chondrocytes. Articular cartilage provides a smooth, low-friction surface and distributes mechanical loads across the joint. It lacks blood vessels and nerves, relying on diffusion from synovial fluid for nutrient exchange. This avascular nature limits its capacity for repair. Synovial joints are further classified based on shape and movement. Hinge joints permit movement primarily in one plane, allowing flexion and extension. Pivot joints allow rotational movement around a single axis. Ball and socket joints permit movement in multiple planes, including rotation. Saddle, condyloid, and plane joints allow varying combinations of movement and stability. The specific structure of each joint reflects a trade-off between mobility and strength. Ligaments are dense bands of collagenous connective tissue that connect bones to other bones across joints. They stabilize joints by limiting excessive movement and guiding normal motion patterns. Ligaments exhibit limited elasticity and are susceptible to injury when stretched beyond their physiological range. Healing of ligaments is generally slow due to limited blood supply. Tendons connect muscles to bones and transmit the force generated by muscle contraction to produce movement. Tendons are composed primarily of parallel collagen fibers aligned to resist tensile forces. Although tendons are not part of the joint itself, their positioning and integrity are essential for joint function and stability. Tendon adaptation occurs in response to mechanical loading, increasing strength and stiffness over time. Menisci and articular discs are fibrocartilaginous structures present in certain joints. They improve joint congruence, distribute load, absorb shock, and enhance stability. The menisci of the knee are crescent-shaped structures that compensate for mismatched bone surfaces. Peripheral regions of these structures have limited blood supply, while central regions are avascular, affecting healing potential after injury. Bursae are small, fluid-filled sacs located near joints where tendons, ligaments, or muscles pass over bone. They reduce friction and cushion movement. Bursae are lined with synovial membrane and contain synovial fluid. Inflammation of bursae can impair movement and cause pain, but does not directly involve the joint cavity. Connective tissues within and around joints adapt to mechanical stress. Regular loading maintains collagen alignment, tensile strength, and joint stability. Reduced movement or immobilization leads to weakening of ligaments, tendons, and joint capsules, resulting in decreased stability and range of motion. Joint development and maintenance depend on coordinated interactions between bone, cartilage, synovium, and connective tissues. During growth, joints shape themselves in response to movement and loading. In adulthood, joint integrity relies on balanced mechanical use, adequate nutrient supply, and intact connective tissues. Joint tissues exhibit limited regenerative capacity. Damage to articular cartilage and fibrocartilage often results in incomplete repair and altered joint mechanics. Progressive degeneration of joint structures can impair mobility and load distribution. The structure and function of joints reflect a balance between durability and mobility, enabling the wide range of movements required for human activity while maintaining skeletal integrity. The muscular system consists of specialized tissues capable of contraction, generating force and movement. Human muscles are responsible for voluntary motion, posture maintenance, joint stabilization, propulsion of substances through organs, and production of body heat. Muscle tissue accounts for approximately 40% of total body mass in an average adult, though this proportion varies with sex, age, and physical conditioning. There are three distinct types of muscle tissue: skeletal muscle, cardiac muscle, and smooth muscle. Each type has unique structural and functional characteristics adapted to specific physiological roles. All muscle cells share the fundamental ability to convert chemical energy into mechanical force through the interaction of contractile proteins. Skeletal muscle is composed of long, cylindrical fibers that are multinucleated and striated due to the organized arrangement of contractile filaments. These muscles are attached to bones via tendons and are under voluntary control through the somatic nervous system. Skeletal muscles enable precise and powerful movements, ranging from fine motor control to large-scale locomotion. Individual muscle fibers can extend several centimeters in length and are among the largest cells in the human body. Cardiac muscle forms the muscular wall of the heart. Cardiac muscle cells are shorter than skeletal fibers, typically contain a single nucleus, and are interconnected by specialized junctions known as intercalated discs. These junctions contain gap junctions and desmosomes that allow rapid electrical signal transmission and mechanical coupling. Cardiac muscle contracts involuntarily and rhythmically, maintaining continuous blood circulation throughout life. Smooth muscle is found in the walls of hollow organs such as the gastrointestinal tract, blood vessels, airways, bladder, and uterus. Smooth muscle cells are spindle-shaped, non-striated, and contain a single nucleus. Contraction is involuntary and regulated by the autonomic nervous system, hormones, and local chemical signals. Smooth muscle contractions are generally slower and more sustained than those of skeletal muscle, and are well suited for regulating internal organ function. Muscle fibers are composed of myofibrils, which are themselves made up of repeating structural units called sarcomeres. Sarcomeres contain interdigitating thick filaments composed primarily of myosin and thin filaments composed mainly of actin, along with regulatory proteins. The highly ordered arrangement of these filaments produces the striated appearance observed in skeletal and cardiac muscle. Muscle organization extends beyond individual fibers. Fibers are grouped into fascicles, which are surrounded by connective tissue layers. These connective tissues transmit force, provide structural support, and allow coordinated contraction across the muscle. Blood vessels and nerves penetrate these layers to supply nutrients, oxygen, and neural control to muscle tissue. Skeletal muscles exhibit considerable functional diversity. Some are designed for endurance and sustained contraction, while others generate rapid, powerful movements. This diversity is reflected in differences in fiber composition, mitochondrial content, capillary density, and metabolic properties. Muscles involved in posture tend to be resistant to fatigue, while muscles involved in explosive actions fatigue more rapidly. Muscle tone refers to the continuous, low-level contraction present in resting muscles. This tone maintains posture, stabilizes joints, and ensures readiness for movement. Muscle tone is regulated by reflex circuits involving sensory receptors and motor neurons, and it persists even in the absence of conscious effort. Muscle tissue has a high metabolic demand. During contraction, muscles consume large amounts of adenosine triphosphate to fuel cross-bridge cycling between actin and myosin. At rest, muscle metabolism contributes significantly to overall energy expenditure. Heat generated as a byproduct of muscle contraction plays an important role in maintaining body temperature. Muscle growth and maintenance depend on mechanical loading, neural input, and adequate nutrient supply. Increased mechanical demand leads to enlargement of existing muscle fibers through increased synthesis of contractile proteins. Reduced use or loss of neural input results in muscle atrophy, characterized by decreased fiber size and strength. Muscle tissue exhibits limited regenerative capacity. Skeletal muscle contains resident stem cells that can repair damaged fibers to some extent, but extensive injury often results in fibrous tissue replacement. Cardiac muscle has minimal regenerative ability, and damage is typically permanent. Smooth muscle retains greater capacity for cell division and repair compared to the other muscle types. The muscular system functions in close integration with the skeletal, nervous, and circulatory systems. Muscle contraction produces movement by acting on bones, is initiated and regulated by neural signals, and depends on blood flow for oxygen and nutrient delivery. The organization and specialization of muscle tissue enable the wide range of movements and physiological functions required for humans responsible for maintaining continuous blood circulation through rhythmic contraction and relaxation. It consists of 4 chambers, 2 atria that receive blood and 2 ventricles that eject blood. The right side of the heart pumps deoxygenated blood to the lungs via the pulmonary circulation, while the left side pumps oxygenated blood to the systemic circulation. Cardiac muscle cells, known as cardiomyocytes, are striated, branched, and connected by intercalated discs containing gap junctions and desmosomes, allowing mechanical strength and rapid electrical coupling. Heart contraction is initiated and coordinated by an intrinsic electrical conduction system. This system generates spontaneous electrical impulses without requiring external neural input, a property known as automaticity. The sinoatrial node, located in the right atrium, serves as the primary pacemaker due to its highest rate of spontaneous depolarization. Pacemaker cells lack a stable resting membrane potential. Instead, they exhibit a gradual depolarization driven by ion fluxes, primarily sodium and calcium influx, and reduced potassium efflux. Electrical impulses from the sinoatrial node spread through the atrial myocardium, causing atrial contraction, and reach the atrioventricular node. The atrioventricular node introduces a brief conduction delay, allowing time for ventricular filling. From there, impulses travel through the bundle of His, down the right and left bundle branches, and into the Purkinje fiber network, which rapidly distributes the electrical signal throughout the ventricular myocardium, producing synchronized ventricular contraction. The cardiac action potential in contractile cells differs from that of skeletal muscle. It consists of distinct phases involving rapid sodium influx, transient potassium efflux, sustained calcium influx through L-type calcium channels, and final potassium-mediated repolarization. The prolonged plateau phase prevents tetanic contraction, ensuring rhythmic pumping. Calcium-induced calcium release from the sarcoplasmic reticulum is essential for excitation-contraction coupling, linking electrical activity to mechanical force generation. The cardiac cycle includes alternating phases of systole and diastole. During diastole, the ventricles relax and fill with blood. During systole, ventricular contraction ejects blood into the pulmonary artery and aorta. Heart valves ensure unidirectional blood flow and open or close in response to pressure gradients rather than muscular control. The opening and closing of valves produce the characteristic heart sounds heard during auscultation. Heart rate and contractile force are modulated by the autonomic nervous system. Sympathetic stimulation increases heart rate, conduction velocity, and contractility through norepinephrine-mediated effects on beta-adrenergic receptors. Parasympathetic stimulation, primarily via the vagus nerve, decreases heart rate by slowing sinoatrial node depolarization. Hormones, temperature, blood ion concentrations, and physical conditioning also influence cardiac performance. Electrical activity of the heart can be measured at the body surface as an electrocardiogram. Distinct waveforms correspond to atrial depolarization, ventricular depolarization, and ventricular repolarization. Changes in waveform shape, timing, or rhythm reflect alterations in conduction pathways, myocardial integrity, or ion balance. The heart operates continuously throughout life, adapting its output to meet metabolic demands. Cardiac output is determined by heart rate and stroke volume, both of which adjust rapidly during exercise, stress, or illness. Despite its endurance, cardiac tissue has limited regenerative capacity, making proper electrical coordination and metabolic support essential for lifelong function. Blood is a specialized connective tissue composed of cellular elements suspended in a liquid matrix called plasma. It performs transport, regulatory, and protective functions essential for survival. Total blood volume in an adult averages 7–8% of body mass and is tightly regulated to maintain adequate circulation and tissue perfusion. Plasma constitutes approximately 55% of blood volume and is composed primarily of water, with dissolved electrolytes, nutrients, gases, hormones, metabolic waste products, and plasma proteins. The major plasma proteins include albumin, globulins, and fibrinogen. Albumin maintains colloid osmotic pressure and serves as a carrier for various substances. Globulins include transport proteins and immunoglobulins involved in immune defense. Fibrinogen is a soluble precursor required for blood clot formation. The formed elements of blood include erythrocytes, leukocytes, and platelets. Erythrocytes, or red blood cells, are the most abundant cellular component. They are biconcave anucleate cells specialized for gas transport. Their shape increases surface area for diffusion and allows flexibility in narrow capillaries. Hemoglobin, the dominant intracellular protein in erythrocytes, binds oxygen reversibly and facilitates carbon dioxide transport. Erythrocyte production occurs in red bone marrow under the control of erythropoietin, a hormone released primarily by the kidneys in response to hypoxia. Leukocytes, or white blood cells, are nucleated cells involved in immune defense and surveillance. They are classified into granulocytes and agranulocytes based on the presence of cytoplasmic granules. Granulocytes include neutrophils, eosinophils, and basophils. Neutrophils are the most abundant leukocytes and are primary responders to bacterial infection, performing phagocytosis and releasing antimicrobial enzymes. Eosinophils participate in defense against parasitic infections and modulate allergic responses. Basophils release histamine and other mediators that promote inflammation and vascular permeability. Agranulocytes include lymphocytes and monocytes. Lymphocytes are central to adaptive immunity and include B cells, T cells, and natural killer cells. B cells differentiate into plasma cells that produce antibodies specific to antigens. T cells mediate cellular immunity, with subsets responsible for helper, cytotoxic, and regulatory functions. Natural killer cells provide rapid, nonspecific defense against virus-infected and transformed cells. Monocytes circulate briefly in blood before migrating into tissues, where they differentiate into macrophages, or dendritic cells that phagocytose pathogens and present antigens to lymphocytes. Platelets, or thrombocytes, are small, anucleate cell fragments derived from megakaryocytes in the bone marrow. They play a critical role in hemostasis by adhering to damaged blood vessel walls, aggregating to form a temporary plug, and releasing factors that promote clot formation. Platelets also release signaling molecules that influence inflammation and tissue repair. Blood contributes to immune function through both innate and adaptive mechanisms. Innate immunity relies on rapid, nonspecific responses involving phagocytic cells, inflammatory mediators, and complement proteins. The complement system consists of plasma proteins that, when activated, enhance phagocytosis, promote cell lysis, and amplify inflammatory responses. Adaptive immunity provides specificity and memory through antigen recognition and clonal expansion of lymphocytes. Circulating immune cells continuously monitor tissues by migrating between blood and lymphatic vessels. Adhesion molecules and chemokines regulate leukocyte movement to sites of infection or injury. Antibodies present in plasma bind antigens, neutralize toxins, and mark pathogens for destruction. Blood also transports cytokines and other signaling molecules that coordinate immune responses across organs. The composition of blood is dynamically regulated. Bone marrow activity adjusts cell production in response to physiological needs, infection, or blood loss. Plasma volume and solute concentrations are controlled by renal, endocrine, and cardiovascular systems. Disruptions in blood composition impair oxygen delivery, immune defense, coagulation, or metabolic balance, demonstrating the central role of blood in maintaining systemic homeostasis. The lymphatic system is a network of vessels, organs, and tissues that maintains fluid balance, supports immune surveillance, and facilitates the transport of lipids and immune cells. It operates in close association with the cardiovascular and immune systems, but follows a unidirectional flow that returns excess interstitial fluid to the bloodstream. Fluid continuously exits blood capillaries into surrounding tissues due to hydrostatic pressure. Most of this fluid is reabsorbed at the venous end of capillaries, but a significant portion remains in the interstitial space. The lymphatic system collects this excess fluid, now called lymph, preventing tissue swelling and maintaining stable blood volume. Lymph is a clear to pale fluid containing water, electrolytes, plasma proteins, lipids, metabolic waste products, and immune cells, primarily lymphocytes. Lymphatic capillaries are blind-ended vessels distributed throughout most tissues. Their walls consist of overlapping endothelial cells that form one-way flap-like openings. These structures allow interstitial fluid, proteins, and particulate matter to enter when tissue pressure rises, while preventing backflow. Anchoring filaments connect endothelial cells to surrounding connective tissue, helping keep capillaries open during tissue swelling. Lymphatic capillaries merge into larger lymphatic vessels with thin walls and internal valves that ensure unidirectional flow toward the heart. Unlike blood vessels, lymphatic vessels lack a central pump. Lymph movement depends on skeletal muscle contraction, respiratory pressure changes, arterial pulsations, and intrinsic contraction of smooth muscle in vessel walls. Flow is slow but continuous. Lymphatic vessels converge into two main ducts. The thoracic duct drains lymph from the lower limbs, abdomen, left thorax, left upper limb, and left side of the head and neck, emptying into the left subclavian vein. The right lymphatic duct drains the right upper limb, right thorax, and right side of the head and neck into the right subclavian vein. This return of lymph to the venous circulation preserves plasma volume and protein concentration. Lymph nodes are small, encapsulated organs located along lymphatic vessels. They function as biological filters that trap pathogens, cellular debris, and foreign particles. Each lymph node contains an outer cortex rich in B lymphocytes, an inner paracortex dominated by T lymphocytes, and a medulla containing macrophages and plasma cells. As lymph passes through nodes, immune cells monitor its contents and initiate immune responses when antigens are detected. Several specialized lymphoid organs contribute to immune function and fluid regulation. The spleen filters blood rather than lymph, removing aged erythrocytes and bloodborne pathogens while serving as a reservoir for immune cells and platelets. Tonsils sample antigens entering through the oral and nasal cavities. Peyer's patches in the small intestine monitor intestinal microbes and contribute to mucosal immunity. The thymus provides the environment for T lymphocyte maturation and selection during early life. The lymphatic system plays a critical role in lipid absorption. Specialized lymphatic capillaries called lacteals are located within intestinal villi. They absorb dietary fats packaged as chylomicrons, which cannot directly enter blood capillaries. These lipid-rich lymph fluids are transported through lymphatic vessels and eventually released into the bloodstream, contributing to systemic lipid distribution. Fluid balance is tightly regulated by the interaction between capillary filtration and lymphatic drainage. When lymphatic function is impaired, fluid accumulates in tissues, resulting in lymphedema. This condition can arise from congenital defects, surgical removal of lymph nodes, infection, radiation damage, or obstruction of lymphatic vessels. The lymphatic system also serves as a primary pathway for immune cell trafficking. Lymphocytes circulate between blood, lymph, and tissues, allowing continuous surveillance for pathogens or abnormal cells. Chemical signals and adhesion molecules regulate this movement, ensuring rapid immune responses while maintaining tissue integrity. Overall, the lymphatic system is essential for maintaining extracellular fluid balance, supporting immune defense, enabling lipid transport, and preserving the stability of the internal environment. Disruption of lymphatic structure or flow compromises immunity, circulation, and tissue health. Blood pressure is the force exerted by circulating blood against the walls of blood vessels, primarily arteries, and is essential for driving blood flow through the circulatory system. It is determined by cardiac output and systemic vascular resistance. Cardiac output depends on heart rate and stroke volume, while vascular resistance is influenced mainly by vessel diameter, blood viscosity, and total vessel length. Arterial blood pressure fluctuates with the cardiac cycle. Systolic pressure corresponds to ventricular contraction and ejection of blood into the arteries, while diastolic pressure reflects arterial pressure during ventricular relaxation. Mean arterial pressure represents the average pressure driving blood into tissues and must be maintained within a narrow range to ensure adequate organ perfusion. Blood vessels exhibit structural and functional specialization that allows precise control of blood flow and pressure. Elastic arteries contain abundant elastin fibers that allow them to stretch during systole and recoil during diastole, helping to maintain continuous blood flow. Muscular arteries distribute blood to specific organs and regulate flow through changes in smooth muscle tone. Arterioles are the primary resistance vessels. Small changes in their diameter produce large changes in vascular resistance and blood pressure. Vascular smooth muscle contraction and relaxation are central to blood pressure regulation. Vasoconstriction increases resistance and elevates blood pressure, while vasodilation decreases resistance and lowers pressure. These changes are controlled by neural, hormonal, and local mechanisms. Smooth muscle tone depends on intracellular calcium concentration, which is regulated by ion channels, receptors, and signaling pathways. The autonomic nervous system provides rapid, short-term control of blood pressure. Sympathetic nerve fibers release norepinephrine, which binds to adrenergic receptors on vascular smooth muscle, causing vasoconstriction in most systemic vessels. Increased sympathetic activity also elevates heart rate and myocardial contractility, raising cardiac output. Parasympathetic influence on blood vessels is limited, but significantly affects heart rate through vagal input. Baroreceptors are stretch-sensitive mechanoreceptors located in the carotid sinuses and aortic arch. They continuously monitor arterial pressure and relay information to cardiovascular control centers in the brainstem. When blood pressure rises, increased baroreceptor firing reduces sympathetic output and enhances parasympathetic activity, lowering heart rate and promoting vasodilation. When pressure falls, the opposite response occurs, restoring pressure toward normal. Hormonal systems provide longer-term regulation of blood pressure and blood volume. The renin-angiotensin-aldosterone system is activated when renal perfusion decreases, blood sodium levels fall, or sympathetic activity increases. Renin release initiates a cascade producing angiotensin II, a potent vasoconstrictor that raises blood pressure and stimulates aldosterone secretion. Aldosterone increases sodium and water reabsorption in the kidneys, expanding blood volume and sustaining pressure. Antidiuretic hormone, released from the posterior pituitary, promotes water reabsorption in the kidneys and causes vasoconstriction at higher concentrations. Atrial natriuretic peptide, secreted by atrial cardiac muscle in response to increased blood volume, opposes the renin-angiotensin-aldosterone system by promoting sodium excretion, vasodilation, and reduced blood volume. Local control mechanisms fine-tune blood flow to individual tissues. Endothelial cells lining blood vessels release vasoactive substances in response to shear stress, oxygen levels, and chemical signals. Nitric oxide induces smooth muscle relaxation and vasodilation, enhancing blood flow. Endothelin promotes vasoconstriction. Metabolic byproducts such as carbon dioxide, hydrogen ions, potassium ions, and adenosine cause local vasodilation in active tissues, matching blood flow to metabolic demand. Blood pressure regulation also depends on blood volume and viscosity. Increased blood volume elevates venous return, stroke volume, and arterial pressure. Hematocrit influences viscosity. Higher viscosity increases resistance and raises pressure. The kidneys play a central role by adjusting water and electrolyte excretion, thereby controlling long-term blood pressure stability. Effective regulation of blood pressure ensures continuous delivery of oxygen and nutrients and removal of metabolic waste. Disruption of vascular control mechanisms, neural regulation, hormonal balance, or renal function leads to sustained abnormalities in blood pressure that compromise organ function and tissue integrity. The respiratory system enables the exchange of gases between the external environment and the internal tissues, supplying oxygen for cellular metabolism and removing carbon dioxide produced by metabolic processes. It consists of conducting airways that transport air and respiratory surfaces, where gas exchange occurs. Air enters the body through the nasal cavity or oral cavity and passes through the pharynx and larynx into the trachea. The trachea divides into the right and left primary bronchi, which enter the lungs and branch repeatedly into smaller bronchi and bronchioles. These airways are supported by cartilage, smooth muscle, and connective tissue that maintain patency and regulate airflow. The conducting zone warms, humidifies, and filters air, but does not participate directly in gas exchange. The respiratory zone begins at the terminal bronchioles and includes respiratory bronchioles, alveolar ducts, and alveoli. Alveoli are thin-walled, sac-like structures that provide a large surface area for diffusion of gases. Each lung contains hundreds of millions of alveoli, collectively creating a gas exchange surface area of approximately 70 square meters in adults. Alveolar walls are composed primarily of two types of epithelial cells. Type I alveolar cells are thin, flattened cells that form most of the alveolar surface and facilitate rapid diffusion of gases. Type II alveolar cells secrete pulmonary surfactant, a phospholipid protein complex that reduces surface tension, prevents alveolar collapse, and decreases the work of breathing. Alveolar macrophages reside within alveoli and remove particulate matter and pathogens. Gas exchange occurs across the respiratory membrane, which consists of the alveolar epithelium, a fused basement membrane, and the capillary endothelium. This membrane is extremely thin, allowing oxygen to diffuse from alveoli into pulmonary capillary blood and carbon dioxide to diffuse in the opposite direction. Diffusion is driven by partial pressure gradients of gases and follows physical laws governing diffusion rates. Ventilation is the process of moving air into and out of the lungs. It is accomplished by rhythmic contraction and relaxation of respiratory muscles. The diaphragm is the primary muscle of inspiration. Its contraction increases thoracic volume and lowers intrapulmonary pressure, drawing air into the lungs. External intercostal muscles assist by elevating the ribs. Expiration at rest is largely passive, resulting from elastic recoil of lung and chest wall tissues, while forced expiration involves active contraction of abdominal and internal intercostal muscles. Pulmonary compliance refers to the ease with which the lungs and thoracic cage expand. It depends on the elastic properties of lung tissue and the surface tension within alveoli. Surfactant increases compliance by reducing surface tension. Airway resistance, influenced by airway diameter and smooth muscle tone, affects the ease of airflow, particularly during expiration. Pulmonary circulation transports blood between the heart and lungs for gas exchange. Deoxygenated blood from the right ventricle enters the pulmonary arteries and flows through pulmonary capillaries surrounding alveoli. After gas exchange, oxygenated blood returns to the left atrium via pulmonary veins. Pulmonary circulation operates at lower pressure than systemic circulation, reducing the risk of fluid leakage into alveoli. Efficient gas exchange requires matching of ventilation and perfusion. Perfusion relationships vary throughout the lungs due to gravity, posture, and regional differences in airway and vascular resistance. Areas with poor ventilation or perfusion contribute less to overall gas exchange, reducing oxygen uptake and carbon dioxide elimination. Respiratory control centers located in the brainstem regulate the rate and depth of breathing. These centers respond primarily to changes in blood carbon dioxide levels and pH detected by central and peripheral chemoreceptors. Oxygen levels play a secondary role under normal conditions. Sensory input from lung stretch receptors and irritant receptors provides additional feedback to adjust breathing patterns. The respiratory system also contributes to acid-base balance by regulating carbon dioxide elimination. Increased ventilation removes more carbon dioxide, reducing acidity, while decreased ventilation allows carbon dioxide accumulation, increasing acidity. This interaction between respiration and blood chemistry is essential for maintaining stable internal conditions. Overall, the respiratory system integrates structural specialization, mechanical ventilation, and precise regulatory control to ensure continuous gas exchange and support cellular metabolism throughout the body. Cellular respiration is the process by which cells convert chemical energy stored in nutrients into usable energy in the form of adenosine triphosphate. It is essential for maintaining cellular structure, driving biochemical reactions, and supporting physiological functions. In humans, cellular respiration is predominantly aerobic, relying on oxygen as the final electron acceptor, though limited anaerobic pathways operate under specific conditions. Glucose is the primary fuel for most cells, although fatty acids and amino acids also contribute to energy production. Cellular respiration occurs in multiple stages, each with distinct biochemical reactions and cellular locations. The process begins with glycolysis, which takes place in the cytosol and does not require oxygen. During glycolysis, one molecule of glucose is converted into 2 molecules of pyruvate, producing a net gain of 2 ATP molecules and reducing equivalents in the form of NADH. Pyruvate produced by glycolysis is transported into mitochondria under aerobic conditions. There, it is converted into acetyl coenzyme A through oxidative decarboxylation. This reaction releases carbon dioxide and generates additional NADH. Acetyl coenzyme A then enters the citric acid cycle, also known as the Krebs cycle, which occurs in the mitochondrial matrix. This cycle consists of a series of enzyme-catalyzed reactions that oxidize acetyl groups completely to carbon dioxide, while transferring electrons to NAD+ and FAD, forming NADH and FADH2. The majority of ATP is produced during oxidative phosphorylation, which occurs along the inner mitochondrial membrane. Electrons carried by NADH and FADH2 are transferred through a series of protein complexes, collectively known as the electron transport chain. As electrons move through these complexes, energy is released and used to pump protons from the mitochondrial matrix into the intermembrane space, creating an electrochemical gradient. This proton gradient represents stored potential energy. Protons flow back into the matrix through ATP synthase, a membrane-bound enzyme that couples proton movement to ATP synthesis. This process is known as chemiosmosis. Oxygen serves as the final electron acceptor at the end of the electron transport chain, combining with electrons and protons to form water. Without oxygen, the electron transport chain cannot function, halting aerobic ATP production. Mitochondria are central to oxygen utilization and energy metabolism. Their number and size vary among cell types depending on energy demand. Muscle cells, neurons, and hepatocytes contain large numbers of mitochondria, reflecting high ATP requirements. Mitochondria possess their own DNA and ribosomes, enabling synthesis of some proteins required for respiratory function, though most are encoded by nuclear genes. Under conditions of limited oxygen availability, cells rely more heavily on anaerobic metabolism. In anaerobic glycolysis, pyruvate is reduced to lactate, allowing regeneration of NAD+ needed to sustain glycolysis. This pathway yields significantly less ATP than aerobic respiration and leads to lactate accumulation, which can alter intracellular and extracellular pH. Lactate can later be transported to the liver and converted back into glucose. Oxygen utilization at the cellular level is tightly regulated. The rate of cellular respiration depends on substrate availability, oxygen supply, and the cell's energy demand. High levels of ADP and inorganic phosphate stimulate oxidative phosphorylation, while high ATP concentrations inhibit key regulatory enzymes. This feedback ensures efficient matching of ATP production to cellular needs. Cellular respiration also generates heat as a byproduct. This heat contributes to maintaining body temperature. Specialized mechanisms, such as uncoupling proteins in brown adipose tissue, allow controlled dissipation of the proton gradient to generate heat instead of ATP, playing a role in thermoregulation. Efficient oxygen delivery by the respiratory and cardiovascular systems is critical for sustained cellular respiration. Impairment of oxygen supply, mitochondrial function, or key enzymes disrupts ATP production, compromising cellular viability and organ function. Cellular respiration, therefore, represents a fundamental link between oxygen intake, nutrient metabolism, and the energy requirements of the human body. The digestive system is responsible for the mechanical and chemical breakdown of food, absorption of nutrients, and elimination of indigestible material. It is organized as a continuous tube extending from the mouth to the anus, with associated accessory organs that secrete enzymes and regulatory substances. Digestive processes ensure that macromolecules are converted into absorbable units that can be transported to cells for energy, growth, and maintenance. Digestion begins in the oral cavity, where food is mechanically fragmented by mastication and mixed with saliva. Saliva contains water, electrolytes, mucus, and enzymes, including salivary amylase, which initiates the digestion of starch into smaller carbohydrates. The tongue assists in bolus formation and swallowing. The pharynx serves as a shared pathway for food and air, directing the bolus toward the esophagus, while protective reflexes prevent entry into the respiratory tract. The esophagus is a muscular tube that transports food to the stomach through coordinated peristaltic contractions. The lower esophageal sphincter regulates entry into the stomach and prevents reflux of gastric contents. The stomach acts as a reservoir and mixing chamber, where food is churned and exposed to gastric secretions. Gastric glands secrete hydrochloric acid, pepsinogen, intrinsic factor, mucus, and bicarbonate. Hydrochloric acid denatures proteins, activates pepsin, and inhibits microbial growth. Pepsin begins protein digestion by cleaving peptide bonds. The stomach lining is protected from acidic conditions by a mucus bicarbonate barrier and tight epithelial junctions. Gastric emptying into the small intestine occurs gradually and is regulated by neural and hormonal signals in response to the composition and volume of gastric contents. The small intestine is the primary site of digestion and nutrient absorption. It consists of the duodenum, jejunum, and ileum. The duodenum receives acidic chyme from the stomach, along with bile from the liver and digestive enzymes from the pancreas. Bicarbonate-rich secretions neutralize gastric acid, creating an optimal pH for enzymatic activity. Pancreatic enzymes digest carbohydrates, proteins, lipids, and nucleic acids into absorbable molecules. The inner surface of the small intestine is highly specialized to maximize absorptive capacity. Circular folds, villi and microvilli, dramatically increase surface area. Enterocytes lining the villi contain brush border enzymes that complete digestion at the cell surface. Monosaccharides and amino acids are absorbed into the bloodstream via specific transporters, while fatty acids and monoglycerides are absorbed into enterocytes, reassembled into triglycerides, and packaged into chylomicrons for transport through the lymphatic system. The large intestine primarily absorbs water, electrolytes, and certain vitamins produced by resident microorganisms. It includes the cecum, colon, rectum, and anal canal. The colon compacts undigested material into feces and stores it until elimination. Smooth muscle contractions move contents through the large intestine at a slower pace than in the small intestine, allowing extensive water reabsorption. Digestive activity is regulated by an extensive enteric nervous system embedded within the gastrointestinal tract wall. This system coordinates motility, secretion, and blood flow independently, though it is modulated by the central nervous system. Hormones such as gastrin, secretin, cholecystokinin, and motilin regulate digestive secretions, gastric emptying, and intestinal motility in response to food intake. Blood supply to the digestive organs supports nutrient transport and metabolic activity. Absorbed nutrients enter the portal circulation and are transported to the liver for processing, storage, or distribution. The digestive system functions continuously, adapting its activity to feeding patterns, diet composition, and physiological state. Effective digestion and absorption depend on the integrity of the gastrointestinal epithelium, coordinated muscle activity, adequate enzyme secretion, and balanced regulatory control. Disruption of any component impairs nutrient availability and compromises overall metabolic function. The liver and pancreas are essential organs for metabolic regulation, digestion, and maintenance of internal chemical balance. They integrate nutrient processing, energy storage, detoxification, and hormonal control, allowing the body to adapt to changing nutritional and metabolic conditions. The liver is the largest internal organ and receives blood from both the hepatic artery and the portal vein. Portal blood carries absorbed nutrients, toxins, and metabolites from the gastrointestinal tract directly to the liver for processing before entering systemic circulation. Hepatocytes, the primary functional cells of the liver, are arranged in lobules with extensive contact with blood-filled sinusoids, enabling efficient exchange of substances. Carbohydrate metabolism is a central function of the liver. Hepatocytes regulate blood glucose concentration by storing excess glucose as glycogen during periods of abundance and releasing glucose during fasting. Glycogenesis, glycogenolysis, and gluconeogenesis occur in the liver under hormonal control. The liver synthesizes glucose from non-carbohydrate precursors such as lactate, glycerol, and amino acids, ensuring a continuous supply for glucose-dependent tissues, particularly the brain and red blood cells. Lipid metabolism is another major hepatic function. The liver synthesizes fatty acids, triglycerides, phospholipids, and cholesterol. It packages lipids into lipoproteins for transport in the bloodstream and removes circulating lipoprotein remnants. The liver converts excess carbohydrates and proteins into fatty acids for long-term energy storage in adipose tissue. It also produces bile acids from cholesterol, which are essential for dietary fat digestion and absorption. Protein metabolism in the liver involves deamination and transamination of amino acids, synthesis of plasma proteins, and detoxification of nitrogenous waste. The liver produces albumin, clotting factors, transport proteins, and many components of the immune system. Ammonia generated during amino acid breakdown is converted into urea through the urea cycle, preventing toxic accumulation and allowing safe excretion by the kidneys. The liver plays a critical role in detoxification and biotransformation. Hepatocytes contain enzyme systems that modify drugs, hormones, and toxins, making them more water-soluble for elimination. These processes often occur in two phases involving oxidation, reduction, hydrolysis, and conjugation reactions. The liver also metabolizes endogenous substances such as steroid hormones and bilirubin, derived from hemoglobin breakdown. Bile production is a key digestive function of the liver. Bile contains bile salts, phospholipids, cholesterol, bilirubin, and electrolytes. Bile salts emulsify dietary fats in the small intestine, increasing surface area for enzymatic digestion. Bile is secreted into bile canaliculi, transported through bile ducts, and stored in the gallbladder between meals. Release into the intestine is regulated by hormonal signals in response to fat intake. The pancreas has both exocrine and endocrine functions. The exocrine pancreas produces digestive enzymes and bicarbonate-rich fluid that are secreted into the duodenum. Pancreatic enzymes include amylases, proteases, lipases, and nucleases, which digest carbohydrates, proteins, fats, and nucleic acids. Bicarbonate neutralizes acidic gastric contents, protecting intestinal mucosa and optimizing enzyme activity. The endocrine pancreas consists of clusters of cells known as pancreatic islets. These cells secrete hormones directly into the bloodstream. Insulin, produced by beta cells, lowers blood glucose by promoting cellular uptake of glucose and stimulating glycogen and lipid synthesis. Glucagon, produced by alpha cells, raises blood glucose by stimulating glycogen breakdown and gluconeogenesis in the liver. Other islet hormones modulate digestive activity and metabolic balance. Metabolic regulation depends on coordinated interaction between the liver, pancreas, and peripheral tissues. Hormonal signals adjust nutrient storage and mobilization based on feeding or fasting states. Disruption of liver or pancreatic function impairs glucose regulation, lipid balance, protein synthesis, and detoxification, demonstrating their central role in maintaining metabolic homeostasis. The gut microbiome consists of trillions of microorganisms that reside primarily in the gastrointestinal tract, with the highest density in the large intestine. These microorganisms include bacteria, archaea, viruses, and fungi. The composition of the microbiome is shaped by genetics, mode of birth, diet, environment, age, and medication exposure, particularly antibiotics. Despite individual variation, the microbiome performs conserved physiological functions essential to host health. Microbial colonization begins at birth and expands rapidly during early life. By adulthood, the gut microbiome reaches relative stability, though it remains responsive to dietary changes and environmental influences. Most gut bacteria belong to a limited number of dominant phyla, with firmicutes and bacteroidetes accounting for the majority, alongside smaller contributions from actinobacteria, protobacteria, and others. Gut microorganisms participate in the digestion of dietary components that are resistant to human digestive enzymes. Complex carbohydrates such as dietary fibers and resistant starches are fermented by microbial enzymes into short-chain fatty acids, primarily acetate, propionate, and butyrate. These metabolites serve as energy sources for colonocytes, contribute to systemic energy metabolism, and influence lipid and glucose regulation in peripheral tissues. The microbiome plays a significant role in vitamin synthesis. Certain gut bacteria produce vitamins, including vitamin K and several B vitamins, which can be absorbed by the host and contribute to nutritional status. Although these microbial contributions may not fully meet daily requirements, they support overall metabolic balance. Gut microbes influence the development and function of the immune system. Early microbial exposure is critical for immune maturation, promoting the differentiation of immune cells and the establishment of immune tolerance. The microbiome helps train the immune system to distinguish between harmless antigens and pathogens, reducing inappropriate inflammatory responses. Microbial signals regulate the production of antimicrobial peptides and the maintenance of immune cell populations within the intestinal mucosa. The intestinal epithelium and microbiome form a functional barrier that limits the entry of pathogens and toxins. Commensal microorganisms compete with pathogenic species for nutrients and attachment sites, a phenomenon known as colonization resistance. They also stimulate mucus production and strengthen tight junctions between epithelial cells, contributing to barrier integrity. Microbial metabolites act as signaling molecules that influence host physiology beyond the gut. Short-chain fatty acids interact with host receptors to regulate inflammation, gut motility, and energy homeostasis. Other microbial products affect bile acid metabolism, altering lipid digestion and cholesterol balance. The microbiome also modulates the bioavailability and activity of certain drugs by metabolizing them before absorption. Communication between the gut microbiome and the nervous system occurs through neural, endocrine, and immune pathways, collectively referred to as the gut-brain axis. Microbial metabolites and signaling molecules can influence enteric neurons, vagal afferents, and central nervous system activity. These interactions affect gastrointestinal motility, secretion, and visceral sensitivity. The composition of the gut microbiome is dynamically regulated by host factors such as diet, immune responses, and intestinal transit time. Dietary patterns strongly influence microbial diversity and metabolic output. High-fiber diets promote microbial populations that produce beneficial metabolites, while diets high in fat and low in fiber are associated with reduced diversity and altered microbial function. Disruption of the normal microbial balance, known as dysbiosis, alters metabolic and immune interactions between host and microbiota. Such imbalances affect nutrient processing, barrier function, and immune regulation. The gut microbiome therefore represents an integral component of human physiology, functioning as a metabolically active system that interacts continuously with host tissues to support digestion, immunity, and metabolic homeostasis. The renal system maintains internal chemical stability by regulating fluid volume, electrolyte concentrations, waste elimination, and blood pressure. It consists primarily of two kidneys, paired ureters, the urinary bladder, and the urethra. The kidneys perform the essential processes of filtration, reabsorption, secretion, and excretion, allowing precise control of the composition of body fluids. Each kidney contains approximately 1 million nephrons, the functional units responsible for urine formation. A nephron consists of a renal corpuscle and a renal tubule. The renal corpuscle includes the glomerulus, a network of capillaries, and Bowman's capsule, which surrounds it. Blood enters the glomerulus under pressure, and water and small solutes are filtered into Bowman's capsule, forming the initial filtrate. Glomerular filtration is driven by hydrostatic pressure and is highly selective. Large proteins and blood cells are retained in the circulation, while water, electrolytes, glucose, amino acids, and metabolic waste products pass into the filtrate. The rate of filtration is regulated by changes in blood pressure, afferent and efferent arterial diameter, and intrinsic autoregulatory mechanisms that stabilize renal function over a range of pressures. After filtration, the filtrate passes through the renal tubule, which is divided into the proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct. Each segment has specialized transport properties. The proximal convoluted tubule reabsorbs the majority of filtered water, sodium, glucose, amino acids, and bicarbonate. This reabsorption is largely active and tightly regulated to prevent loss of essential substances. The loop of Henle establishes an osmotic gradient in the renal medulla through a countercurrent multiplier system. The descending limb is permeable to water but not to solutes, while the ascending limb actively transports sodium and chloride but is impermeable to water. This arrangement allows the kidneys to concentrate or dilute urine depending on the body's hydration state. The distal convoluted tubule and collecting duct fine-tune urine composition. Reabsorption of sodium, water, calcium, and bicarbonate, and secretion of potassium and hydrogen ions occur here under hormonal control. Aldosterone increases sodium reabsorption and potassium secretion, while antidiuretic hormone increases water permeability of the collecting ducts, allowing more water to be reabsorbed and producing concentrated urine. The kidneys play a central role in waste filtration and excretion. Nitrogenous waste products such as urea, creatinine, and uric acid are removed from the bloodstream and eliminated in urine. These substances are generated from normal metabolic processes and must be continuously cleared to prevent toxic accumulation. Acid-base balance is regulated by renal handling of hydrogen ions and bicarbonate. The kidneys excrete excess hydrogen ions and generate new bicarbonate, helping maintain blood pH within a narrow range. This renal regulation complements respiratory control of acid-base balance and is essential for normal enzyme function and cellular metabolism. Renal function is closely linked to blood pressure regulation. The kidneys adjust extracellular fluid volume by controlling sodium and water excretion. Specialized cells in the juxtaglomerular apparatus sense changes in blood pressure, sodium concentration, and sympathetic activity. In response, they release renin, initiating hormonal cascades that influence vascular resistance and fluid balance. The kidneys also have important endocrine functions. They produce erythropoietin, which stimulates red blood cell production in the bone marrow, and activate vitamin D to its biologically active form, which is essential for calcium and phosphate balance. These hormonal actions integrate renal activity with skeletal, cardiovascular, and hematologic systems. Urine formed by the kidneys flows through the ureters to the urinary bladder, where it is stored until elimination. Micturition involves coordinated neural control of bladder muscle contraction and urethral sphincter relaxation. Proper renal function ensures efficient waste removal, stable internal chemistry, and long-term maintenance of physiological homeostasis. Fluid, electrolyte, and acid-base balance are essential for maintaining cellular function, enzyme activity, and overall physiological stability. These balances depend on coordinated regulation by the kidneys, respiratory system, cardiovascular system, and endocrine signaling. Small deviations can disrupt membrane potentials, metabolism, and organ function. Body fluids are distributed between intracellular and extracellular compartments. Approximately 2/3 of total body water is intracellular, while the remaining third is extracellular, divided into interstitial fluid and plasma. The movement of water between compartments is governed by osmotic gradients created by solute concentrations, primarily electrolytes and plasma proteins. Electrolytes are charged ions dissolved in body fluids that participate in electrical signaling, muscle contraction, and chemical reactions. Sodium is the principal extracellular cation and a major determinant of extracellular fluid volume and osmolality. Potassium is the principal intracellular cation and is critical for maintaining resting membrane potential and normal neuromuscular function. Calcium, magnesium, chloride, bicarbonate, and phosphate also play essential roles in structural integrity, signaling, and metabolic regulation. Electrolyte balance is achieved through regulated intake, distribution between compartments, and excretion. The kidneys are the primary organs responsible for maintaining electrolyte concentrations by adjusting reabsorption and secretion along the nephron. Hormones such as aldosterone, antidiuretic hormone, parathyroid hormone, and atrial natriuretic peptide modulate renal handling of sodium, potassium, calcium, phosphate, and water. Fluid balance depends on the relationship between intake and output. Water is gained through ingestion and metabolic processes and lost through urine, sweat, respiration, and feces. The sensation of thirst and the release of antidiuretic hormone are triggered by changes in plasma osmolality and blood volume, promoting water intake and conservation when needed. Acid-base balance refers to the regulation of hydrogen ion concentration in body fluids, which determines pH. Normal arterial blood pH is maintained within a narrow range compatible with protein structure and enzyme activity. Acids are continuously produced by metabolism, including carbonic acid from carbon dioxide and non-volatile acids from protein and phospholipid metabolism. Buffer systems provide immediate resistance to changes in pH. The bicarbonate buffer system is the most important in extracellular fluid, involving a reversible reaction between carbon dioxide, carbonic acid, hydrogen ions, and bicarbonate. Phosphate buffers and protein buffers, including hemoglobin, also contribute to pH stability. The respiratory system regulates acid-base balance by controlling carbon dioxide elimination. Increased ventilation removes carbon dioxide, reducing hydrogen ion concentration and raising pH. Decreased ventilation allows carbon dioxide to accumulate, increasing acidity. Respiratory compensation occurs rapidly in response to acid-base disturbances. The kidneys provide long-term regulation of acid-base balance. They excrete hydrogen ions, reabsorb filtered bicarbonate, and generate new bicarbonate to replace that consumed by buffering acids. Renal tubular cells use specialized transporters and enzymes to manage acid secretion and bicarbonate handling, adjusting urine acidity according to systemic needs. Fluid, electrolyte, and acid-base regulation are interdependent. Changes in electrolyte balance influence water distribution and acid-base status, while acid-base disturbances affect electrolyte binding and transport. For example, hydrogen ion concentration alters potassium distribution between intracellular and extracellular compartments. Maintaining these balances ensures stable cell volume, effective nerve impulse transmission, coordinated muscle contraction, and optimal enzymatic reactions. Failure of regulatory mechanisms leads to dehydration or fluid overload, electrolyte imbalances, and acid-base disorders that impair organ function and threaten physiological stability.